Hi everyone, today we're going to be solving AQA GCSE Biology Higher Tier Paper 1H. In this particular video, we are solving Specimen 2018 question paper. In this video, we'll be solving the entire question paper. Plants transport minerals and water from the root to the leaves. Plants move mineral ions from a low concentration in the soil to a high concentration in the root cells. What process do plants use to move these mineral ions into root cells? So, plant uses active transport to move mineral ions into the root cells. Now, the reason why the plant uses active transport is because the concentration of mineral ions is higher, in the root cells than actually the surrounding soil. So, from the soil to the root cell, it is like going uphill. It is against the concentration gradient. It is like going uphill. So, it requires energy. Active transport uses the energy from respiration to pump ions against the concentration gradient. Whereas, let's see the other option. Diffusion is the movement down the concentration gradient. Evaporation is actually when water turns into water vapor, and osmosis is the movement of water from a region of high water potential to a region of low water potential, or from a region of high water concentration to a region of low water concentration. Describe how water moves from roots to the leaves. Water moves from the roots to the leaves by transpiration pool in the xylem. Plants lose water through the stomata in the leaves. The epidermis can be peeled from a leaf. The stomata can be seen using a light microscope. Table 1 shows the data a student collected from 5 areas of one leaf. We can see the upper surface and the number of stomatas are given. For leaf 1, it is 3, and lower surface it is 44. We can see the lower surface has more number of stomata. For leaf 2, there is no stomata on the upper surface, but the lower surface has 41 stomata. And then, the student repeats the process, and then the student calculates the average number of stomata for the upper surface and finds it to be 2, and the lower surface is not done, so we'll have to do it. Describe how the student might have collected the data in table 1. Obviously, the student is collecting data about the number of stomata in the upper surface and the lower surface in 5 areas of on one leaf. So what the student can do is the student can mount the epidermis on a slide and then count the number of stomata in one area and then repeat in 4 more areas and repeat the method on the other surface of the leaf. Then the student can calculate the average. What is the median number of stomata on the upper surface of the leaf? So on the upper surface of the leaf, we can see the number ranges from 1 to 5. So, alright, so if we write down the numbers in a serial, 0 and then 1, 1, 3 and 5, we can see the middle number is 1. So the answer will be 1. The median number of stomata is 1 on the upper surface of the leaf. Calculate the value of x in table 1. Give your answer to two significant figures. So the value of x will be the average of the stomatas in the lower surface. So we're going to add 44 with 41 plus 40, 42 along with 39, and we're going to divide it by 5. This gives us 41.2. However, the answer must be given in two significant figures. So the mean number of stomata on lower surface of the leaf will be 41. The plant used in this investigation has very few stomata on the upper surface of the leaf. Explain why this is an advantage to the plant. Having less stomata on the upper surface means the plant will lose less amount of water as water vapor, and so it will not wilt easily. Tobacco mosaic virus TMV is a disease affecting plants. Figure 1 shows a leaf infected with TMV. Yellow patches where TMV has destroyed chloroplast. All tools should be washed in disinfectant after using them on plants infected with TMV. Suggest why. Since it is a virus, the purpose will be to kill the virus and to prevent the virus from spreading. Scientists produced a single plant that contained a TMV-resistant gene. Suggest how scientists can use this plant to produce many plants with the TMV-resistant gene. The scientist can take stem cells from the meristem of that TMV-resistant plant and then use tissue culture to grow that TMV-resistant plant and multiply in number. So guys, writing the part, scientists can take stem cells from the meristem, or the scientist can use tissue culture. Both of them, you can write any one of them, score the full marks. Some plants produce fruits which contain glucose. Describe how you would test for the presence of glucose in fruit. Since glucose is a reducing sugar, so we can hit the solution that we can get, the juice that we can get from the fruit, we can heat that solution, and we can heat it with Benedict solution. Once we heat it with Benedict solution, the glucose will turn the Benedict solution from blue to orange, indicating presence of glucose. TMV can cause plants to produce less chlorophyll. This causes leaf discoloration. Explain why plants with TMV have stunted growth. Since plants with TMV, they have less amount of chlorophyll, they are less able to photosynthesize, and as a result they will produce less amount of glucose, so less amount of energy is released for growth, because glucose is also needed for respiration as well as for growth. So glucose is also needed for making amino acids, which makes protein, and proteins are needed for growth of the plant. Glucose is also needed for making cellulose, and cellulose are needed for the cell wall formation for the plant cells. Microorganisms cause infections. The human body has many ways of defending itself against microorganisms. Describe 2 ways the body prevents the entry of microorganisms. This particular question has 4 marking points. However, the examiner has given us only 2. So we are gonna be able to write only 2 of the 4 marking points that I am mentioning here. First of all, we need to understand the skin forms a barrier, which also produces antimicrobial secretions in the skin, which prevents bacteria from entering our body. Our hair in the nose traps particles which may contain pathogen. Our trachea or bronchi has mucus which traps pathogen, and it also contains enzymes that kills the pathogen. We have acid in our stomach that kills pathogens in food as well. In 2014, the Ebola virus killed almost 8,000 people in Africa. Drug companies have developed a new drug to treat Ebola. Explain what testing must be done before this new drug can be used to treat people. Before a drug can be used for treating people, it must go through preclinical trials. First of all, the preclinical trials will be done on cells or tissues and live animals to test for toxicity, dosage, and its efficacy. Then clinical trials can be done on healthy volunteers and patients at a very low dose to understand its side effects. And then only we can then do trials to find the optimum dosage and test for the efficacy of that particular drug. After that, the drug can go through a double-blind trial by using placebo. When using placebo, it doesn't contain the new drug, and random allocation of patients to groups are done where placebos are given instead of the new drug. And a peer review of the data is done to help prevent false claim of a particular medicine. All living cells respire. Respiration transfer energy from glucose for muscle contraction. Describe how glucose from small intestine is moved to muscle cell. So glucose from small intestine is absorbed by diffusion into the bloodstream because glucose is at higher concentration in the small intestine. Then the glucose is then delivered into the bloodstream. Glucose is absorbed by diffusion into the bloodstream. Once the glucose is absorbed by diffusion into the bloodstream, then the blood delivers the glucose to the muscles in the capillaries. Figure 2 shows an experiment to investigate anaerobic respiration in yeast cell. We can see that we have a boiling tube with the liquid paraffin, and underneath we have yeast cell in sugar solution in tube A. The tube A is connected and by using a rubber bung sealed so that the gas produced flows through tube B, where the gas is bubbled through a indicator solution, and the gas bubbles, as you can see, can escape through this particular tube. What is the purpose of the liquid paraffin in tube A? We know that to observe anaerobic respiration in yeast cell, we must prevent oxygen from coming in. So the liquid paraffin is there to prevent oxygen from getting into the solution. Point 1, to prevent evaporation. Well, that is not the purpose. To stop air from getting in, yes, we don't want air to get in because air contains oxygen. To stop the temperature from going up, no. To stop temperature changing, we can use water bath. However, that is not the goal of the experiment. And to stop water from getting in, since water is heavier than oil or paraffin, so it will sink down. So that is also not the purpose of this particular experiment. So we want to stop the air from getting in so that it doesn't bring in oxygen, and the oxygen will then contaminate the experiment. The indicator solution in tube B shows changes in the concentration. The indicator solution in tube B shows changes in the concentration of carbon dioxide. The indicator is blue when the concentration of CO2 is very low, and green when the concentration of CO2 is just low, and yellow when the concentration of CO2 is high. What color would you expect the indicator to be in tube B during maximum rate of anaerobic respiration? since maximum rate of anaerobic respiration is going on, and when glucose is broken down in anaerobic respiration by yeast, it produces a large quantity of CO2, so it will go yellow. Suggest how the experiment could be changed to give a reproducible way to measure the rate of the reaction. Include any apparatus that you would use. So to replicate the experiment in a way so that we can measure the rate of the reaction, we can do two things. First of all, we can collect the CO2 using a gas syringe, and then because we're trying to find out the rate, we can calculate the amount of the volume of CO2 collected within a particular time period by using a timer. Compare the anaerobic respiration in yeast cell with anaerobic respiration in a muscle cell. So in a yeast cell, ethanol is produced, but muscle produces lactic acid. In a yeast cell, carbon dioxide is produced, but muscles does not produce carbon dioxide. Both yeast cell and muscle cells release small amount of energy when they do anaerobic respiration. A student investigated the effect of different sugar solution on a potato tissue. This is the method used. 30 cm cube of 0.8 mol per dm cube sugar solution is added to a boiling tube. Repeat step 1 with equal volumes of 0.64 and 0.2 mol per dm cube sugar solutions. Then use water to give a concentration of 0.0 mol per dm cube. So we can see this is the control. Cut 5 cylinders of potato of equal size using a cork borer. Weigh each potato cylinder and place one in each tube. Remove the potato cylinder from the solutions after 24 hours. Dry each potato cylinder with a paper towel. Reweigh the potato cylinders. So we can see the concentration of the sugar solution from 0 to 0.8, and we can see the starting mass, and as the concentration increases, alright, okay, so we can see the final mass, alright, as the concentration increases, the final mass decreases regardless of the starting mass. And then the change in percentage, we can see, all right, as the concentration increases, the change in percentage mass goes negative, meaning water is lost. In the beginning, water is gained. In this one, water is gained, and as we go down up until here, water is gained, and at 0.6 mol per dm cube, water is lost, and at 0.8 mol per dm cube, more water is lost. Now the question says, calculate the value of x in table 2. So to calculate the value of x, we can see it is about percentage change. So let's see what is the percentage change in mass. The original value was 1.35. So we have a change in mass of 0.15. We will divide it by 1.35 into 100%. And when we do that, we get a percentage change in mass of 11.1%. Whenever it is not mentioned that what significant figure to use in the exam, always go for 3 significant figure, because our exam significant figure is always 3 significant figure. Why did the student calculate the percentage change in mass as well as the change in grams? The student wanted the results to be compared, or we can say they had different masses at the start of the experiment so that the results can be compared. Complete figure 3 using the data from table 2. Choose a suitable scale and label for the x-axis and plot the percentage change in mass, and then we'll have to draw a line of best fit. So first of all, we will have to do a, for example, we have to choose a suitable scale and label the x-axis. X-axis is always our independent variable, so it will be concentration of sugar solution. The concentration of sugar solution in mol per dm cube. Now we will have to choose a suitable scale. So we will go back to our graph and see what is the maximum concentration. The maximum concentration is 0.8. So we can start with 0.2 on each scale, 0.4, 0.6, and 0.8 respectively. And finally, the last one is 1.0. However, we do not have any value for that. Use your graph in figure 3 to estimate the concentration of the solution inside the potato cells. To estimate the concentration of solution inside the potato cell, we must look at the line where it crosses the x-axis. The line where it crosses the x-axis, the concentration of inside the potato cells and the outside the potato cells are equal. So at that particular place, our reading is 0.46. So 0.46 mole protein cube. Guys, do not worry, the answer is actually taken from your graph. So even if your graph is wrong, you will still get this particular answer correct as long as you do it from the place where the graph crosses where the line crosses the x-axis. The results in table 2 shows the percentage change in mass of the potato cylinders. Explain why the percentage change results are positive and negative. From 0.0 to 0.4, water moves into the cells of the potato, and from 0.6 to 0.8, water leaves the cells of the potato. So the water enters the potato cells by osmosis and leaves the potato cell by osmosis. Guys, when the question says that the question asks about explanation for the percentage change in results, this should be the particular explanation. So for every step, you will say from 0.0 to 0.4, water moves into the potato cells by osmosis. Then for the next step, you will say from 0.6 to 0.8, water leaves the potato cell by osmosis. We will get mark for by osmosis only once, but however, we will have to write it twice. Suggest two possible sources of error in the method given in page 16. So the two possible sources of error in the method given in page 16, if we analyze this particular process, we see that there can be a error in doing the concentration that the student has provided, like 0.8, 0.6, 0.4 and 0.2. And then also there can be a problem with the drying the chips using paper towel. Either excess water or less amount of water will be removed, and then that can affect the mass. Another way of getting wrong is that there can be accuracy. All right, the accuracy of the balance can also matter in getting a correct result. Question 6 talks about human digestive system. We can see the human digestive system represented here. Label the stomach and pancreas on figure 4. And the pancreas will be right underneath the stomach. It's a leaf-like structure. Many people suffer from stomach ulcers caused by a species of bacteria called Helicobacter pylori. The stomach is lined with a protective lining of mucus. Helicobacter pylori are acid-tolerant bacteria which can damage this mucus lining. Suggest how an infection with Helicobacter pylori might result in a stomach ulcer developing. First of all, the bacteria are not killed by the stomach acid, so they can damage the mucus lining. Once the bacteria damages the mucus lining, the stomach acid, hydrochloric acid, then can damage the stomach tissues, which can then lead to ulcer. Helicobacter pylori can also cause stomach cancer. Describe how a person infected with Helicobacter pylori could also develop liver cancer. For example, if the cancer in the stomach is malignant, the cancer cells can spread to other organs via the blood, forming a secondary tumor in the liver. By the way, guys, this process is known as metastasis, and it can basically spread cancer to all parts of the body. Liver is just an example in this case. Gluten is a form of protein found in some grains. Describe the test you would use to find out if protein is present in food. To test for protein, we can add biorate reagent to food samples. If the food sample solution becomes purple in color, that means protein is present. Celiac disease is a disease of the digestive system. It damages the lining of the small intestine when food that contains gluten are eaten. When people with celiac disease eat food that contain gluten, their immune system forms antibodies to gluten. These antibodies then attack the lining of the small intestine. This causes inflammation in the intestine and damages the villi. Symptoms of celiac disease include poor growth. Suggest why a person with celiac disease might have this symptom. symptom. Since a person with celiac disease will have damage to villi, the damage to villi reduces surface area for absorption of food molecules. Therefore, fewer amino acids and glucose are absorbed into the bloodstream, and with less glucose transferred, for energy for respiration is also reduced. And since there are fewer amino acids available, there will be the fewer amino acid available for the person to build new proteins, which will in turn affect their growth. A gardener is looking at the plants in his greenhouse. Some of the plants have a disease. Give two ways the gardener could identify the pathogen infecting the plants. First of all, the gardener can compare them with pictures in a gardening manual or in a website. And then, if he is unable to detect it, then he can do a second process, is that he can take samples and send it to the laboratory for testing. Plants can become unhealthy if they do not have essential mineral ions. Describe the appearance of plants with nitrate deficiency, magnesium deficiency. So, for example, if a plant has nitrate deficiency, the plant will have stunted growth. If the plant has magnesium deficiency, then the leaves will become yellow. Guys, the reason why nitrate deficiency causes stunted growth. Nitrate is needed for making amino acids, and without amino acids, the plants are unable to make protein. So, for example, important proteins, those that are needed for growth, are unable to be made, and as a result the plant have stunted growth. And magnesium deficiency, the leaves become yellow because magnesium is an essential element needed for making chlorophyll. So if the plant leaf decreases in chlorophyll, then we will see the leaves will become yellow. Plants need other mineral ions. Potassium ions are needed for healthy root growth. Phosphate ions are needed for healthy flowers and fruits. The gardener makes his own garden compost. The percentage of mineral in his compost was compared with two fertilizers he could buy. So we can see percentage mineral content. In terms of nitrate ions, the garden compost consists of 0.5, fertilizer S 5.0, and fertilizer T contains 3.0. So nitrate content is highest in fertilizer S. Then for phosphate ion, we can see fertilizer T is highest in concentration. And then for potassium ion, we can see fertilizer S is highest in concentration. And in terms of cost, we can see the garden compost is totally free, however provides all of the nutrients. The gardener buys fertilizer S. Explain why he chooses fertilizer S. Since the plant need nitrate for making protein, and potassium for its general root growth, and phosphate for its general growth, healthy flowers and fruits. So fertilizer S has all of them in high proportion, in comparison with the cost. So we will give a one by one breakdown. Fertilizer has the most nitrogen for good growth and has the highest potassium content for stronger roots. It is also cheaper than fertilizer. However, the fertilizer S lacks phosphate ions. There is less amount of phosphate ion than fertilizer T, although more than compost. So flowers and fruits perhaps are less important for the gardener. Lungworm is an infection. Lungworm can kill dogs. It is caused by a small worm. Figure 5 shows the cycle of the lungworm. Lungworm reproduces in the dog and infects the lungs. The larva, young stages of the worm, are coughed up from the lungs and swallowed. Worm larvae pass out in feces and are picked up by snails. Dogs touch or eat snails, and then that's how they get the lungworm. What type of organism is represented by the snail in the life cycle of the lungworm? We can see the organism is a carrier of a lungworm back into a new dog. That means it is a vetted, because it by itself does not get affected. However, it can pass on the lungworm to a healthy dog. A fungus is not the correct answer, because fungus is basically an organism which so the snail is clearly not a fungus, however, and it is not clearly not a protist. It is a snail is an animal, so cannot be fungus or protist. However, the snail by itself doesn't cause the disease, so it is not a parasite as well. Suggest how the spread of the lungworm disease can be prevented. First of all, we can isolate the infected dogs, or we can treat the infected dogs with anti-lungworm medications. We can educate the owner about picking up their dog feces so that the cycle doesn't continue. Or we can destroy the snails in the surrounding vicinity where the dog moves around. Malaria is a disease spread by mosquitoes. Describe 2 ways to control the spread of malaria. Since malaria is spread by mosquitoes, so if we can prevent people from getting bitten by mosquito, and if we can, for example, we can use mosquito nets, and then we can also, stop the mosquito from breeding by destroying their breeding sites. Figure 6 shows photographs of some animal cells at different stages during their cell cycle. We can see at cell stage A. So stage A looks like that these cells are, they're turning into 2 new cells. In stage B, we can see metaphase. So in A, we can see cytokinesis. In B, we can see metaphase. And in C, we can see the cells like normally. So we can say either it is prophase or it is like the normal cell cycle. The normal, all right, where it is not going through mitosis, just general life cycle of the cell. Which photograph shows cells that are not going through mitosis? So definitely the answer will be C. Describe what is happening in photograph A. So in photograph A, since the process of cytokinesis is going on, so cytoplasm and cell membrane are dividing to form 2 identical daughter cells. A student wanted to find out more about the cell cycle. The student made a slide of an onion root. She counted the number of cells in each stage of the cell cycle in one field of view. Non-dividing cells 20, stage 1, 9, stage 2, 4, and then, the total number of cells that she counted was 36. Each stage of the cell cycle takes different amount of time. Which stage in table 4 is the fastest in the cell cycle? Give a reason for your answer. So stage 4 is the fastest. How could we conclude? Because only one of the cell was seen in stage 4. So must be a very fast cycle at that particular, must be a very fast stage of the cell cycle. That's why only very little amount of cells were caught in that particular stage. So stage 4. And the reason is only one cell seen in this stage. The cell cycle in an onion root tip takes 16 hours. Calculate the length of time stage 2 lasts in typical cells. Give your answer to 2 significant figures. Because stage 2 has 4 cells, so we will calculate based on that, information. 4 divided by total number of cells, which is 36, times 16 into 60. This will give us the answer in minutes, because the answer is required in minutes, which gives us 106.7, which is equivalent to 107 into significant figure. Which is equivalent to 110 into significant figure. Bacteria such as E. coli undergo cell division similar to mitosis. Figure 7 shows a growth curve for E. coli grown in a nutrient broth. So we can see time in hours as the time gets passed, and the logarithmic number of cells, live cells. In the beginning, we can see a lag phase. The number is not increasing. Then we can see a log phase in P, and for Q, we can see a stationary phase. What type of cell division caused the change in number of E. coli from cells at P? So definitely we will say binary fission. Bacteria divides by binary fission. E. coli is a bacteria. Suggest why the number of cells level out at Q. The number of cells may have leveled out due to shortage of nutrients, oxygens, or the death rate became equal to the rate of cell division. Explain how the human circulatory system is adapted to supply oxygen to the tissues and remove waste product from the tissues. This particular question has many points to be written. However, we can pick and choose 6 to 7 points to write a complete answer. And since this question is asked repeatedly in every year in the same manner, so you can give the same answer for all of them. The human circulatory system is a double circulatory system, which means it has a higher blood pressure and a greater flow of blood to the tissues. The heart is made of specialized cardiac muscles which have long protein filaments that can slide past each other to shorten the cell to bring about contraction for pumping the blood. The heart pumps the blood to the lungs in pulmonary arteries so that oxygen can diffuse into blood from air in the alveoli. The blood then returns to the heart via pulmonary vein where heart muscles pump the blood to the body via the aorta. Now oxygen is then carried by specialized cells For example, red blood cells, all right, which contains hemoglobin for carrying the oxygen, and it doesn't have nucleus, so there are more available space to carry the oxygen. The red blood cell carries the the arteries carries the oxygenated blood to tissues where capillaries deliver oxygen to cells for respiration and energy release. The thin walls of the capillary allow faster diffusion of oxygen into the cells. Large surface area of the capillary also maximizes the exchange of CO2 and oxygen. The waste product, example carbon dioxide, diffuses from the cells into the blood plasma, and the waste is then carried, since the blood goes back to the heart in veins, which then have valves to prevent backflow. The cardiac output can vary according to the demand of the particular person. If the person is exercising, the cardiac output increases to remove the waste carbon dioxide and supply the cells with more oxygen. Now guys, this particular question is many points long. You may pick and choose point from here. However, try to include point from all segments, as in get from the first 3 points at least try to mention 1 point. For the second 3 points at least try to mention 2 points, and for the last 5 points at least try to mention like 3 points or 4 points. Okay, I'm going to give you the whole answer. You can pick and choose and write your own answer from these particular points. Monoclonal antibodies are used to measure the levels of hormone in the blood. Pregnant women produce the hormone HCG. HCG is excreted in urine. Figure 8 shows 4 pregnancy test strips. We can see a control window and we can see a result window with A, B, C, D. Alright. Positive test result, a line appears in the control window and the result window. Negative test result, line appears only in the control window. Invalid test result, no line appears in the control window. Which test strip shows a negative test result? So, a negative test result is when we can see that a line appears only in the control window. So, A will be the correct answer. B is not correct answer because it is an invalid test. It doesn't contain any lines. C is a positive test because lines appears in both of the windows. Whereas D has a line only in the result window. So it doesn't mean anything. Alright. If the negative test will only be applicable if the line appears in the control window. Okay, so D is also a invalid test because line appears in the result window but doesn't appear on the control window. Monoclonal antibodies are used for pregnancy testing. Give one other use of monoclonal antibodies. Monoclonal antibodies can be used for locating blood clots and also can be used for locating specific molecules and hormones. Monoclonal antibodies can be also used for treating some cancers as well. Figure 9 shows the part of a pregnancy test strip. We can see the control window, immobilized antibodies specific to the mobile antibodies from the reaction zone. Result window, immobilized antibodies specific to HCG here is present. Reaction zone, there are mobile antibodies specific to HCG here, and these antibodies can move and have the blue dye attached to them. Urine is applied on this particular portion. The pregnancy test strip will show a positive test result when a woman is pregnant. Explain how the pregnancy test strip works to show the positive result. So once the urine is applied in this particular region, the urine first passes through the reaction zone. Once the urine passes through the reaction zone, the urine, if it contains HCG, the HCG will then bind with the HCG binding antibodies. The HCG, those that gets bind with the antibodies, will be left behind here, alright, and the antibodies, those that are free, are now traveling up along with the urine, and the HCG that did not bind with the antibodies will then travel up and bind with the immobilized HCG antibodies, and then a result will appear on the result window. And any antibodies that did not bind at this particular, the mobilized antibody that did not bind at this location, will travel up and enter the control window, where immobilized antibodies that are specific to mobile antibodies are present, and they will react and make an align appear. This will indicate that the test is complete and the test is correct. So the answer can be written like this: as the urine passes passes through the reaction zone. HCG hormone binds to the mobile HCG antibody in the reaction zone. The HCG that did not bind with the mobile antibody then are passed, to the immobilized HCG antibodies in the result zone, and they bind with the antibodies which do not attach, and the antibodies which do not attach to the HCG, they bind to the antibodies in the control zone, and as a result blue dye appear in both control and result zone to show positive result. Guys, thank you for watching the video. Guys, question paper like this will be uploaded every day. Subscribe to the channel and let your friends know about the channel as well. Guys, best of luck for your exam and see you in the next video, guys. Bye bye.